Okay, it is time for the lab check, so make sure that you have completed all of the sections in your lab manual and that you thoroughly understand the material. This lab check has 15 questions and it is worth 20 points. Question number 1. What is the end product of light-dependent reactions involving chlorophyll? Question 2. Predict the relationship between light intensity and this product. Back from question number 1. What's the relationship between that product and light intensity? Question 3. Write your hypothesis. Whatever you had in your lab manual, what was your hypothesis? Question 4. What was your independent variable? Question 5. What was your dependent variable? Question 6. What were your standardized variables? And notice that this question is worth 2 points. There are lots of things that you should have had standardized, but you need to list at least 2 of those standardized variables. Question 7. What caused more and more leaf discs to rise as the experiment progressed? Question 8. Did you record your data in the table in your lab manual? Question 9. Did you complete a line graph of the data from your experiment? And like I told you during the lab, this is worth 4 points. Question 10. What process does this equation represent? Question 11. Describe the relationship between photosynthesis and cellular respiration. Okay, this question is worth 2 points. It's a little bit deeper, so be thorough in your answer. Okay, don't just scribble down a quick answer. Think through it and write down a good, well thought out answer. Question 12. Predict what will happen if you place the control group in the dark after those leaf discs have risen to the top. Question 13. Was your hypothesis supported or falsified? And you do need to use data to support your answer. Don't just say yes or no, but support your answer. Question 14. What can you summarize regarding the rate of photosynthesis and the factor you were testing? In my experiment, I was testing a temperature, but again, you might have chosen a different factor. What can you summarize regarding your factor and the rate of photosynthesis? And finally, question 15, what steps would you recommend to improve the experiment that you performed? Okay, those are all of the questions on the lab check, so make sure that you have written down your final answer for each question, and let's review the lab check. Question number 1. What is the end product of the light-dependent reactions involving chlorophyll? Well, that is oxygen. Question number 2. You were asked to predict the relationship between light intensity and that product, which was oxygen. Well, as light intensity increases, the amount of oxygen produced also increases. Remember that enzymes play an important role in photosynthesis. Okay, they catalyze the photosynthetic reactions, allowing these reactions to occur at a faster speed. But realize also enzymes are affected by many different factors. They're affected by pH, they're affected by heat, they're affected by different types of chemicals. Anything that affects those enzymes that are catalyzing the reaction are also going to affect the rate of the reaction. So realize that's the other question that was asked in your lab manual. I didn't ask you about that, but you should have put something down about those enzymes affecting the rate of the reaction as well. Question number 3, write your hypothesis. Now the answers could vary for this. Again, you might have chosen a different factor than I did. If you went with my demonstration, the best photosynthetic rate will take place in hot water. That's the hypothesis that I began with because I was using temperature as my factor. For question number 4, the independent variable, again, answers may vary for this. For my example, temperature was the independent variable. And just a quick review, remember the independent variable is the factor that varies among the different groups. Then question 5, the dependent variable, well, it is dependent upon that independent variable. And in my experiment, the dependent variable was the amount of time that the leaf discs take to rise. And that was dependent upon the temperature of the solution that they were in. Question number 6, what were the standardized variables? Again, you could have put several different things for this answer. The same amount of solution, okay, you filled up the beakers to the same depth, the same type of solution with the sodium bicarbonate, the same type of leaf. Hopefully you used ivy in all of your cups, or you used spinach leaves in all of your cups. You didn't have one type in one and another in a different cup. Same distance from the light source, okay, unless of course intensity was your factor. Any of those, you may have thought of other standardized variables as well. Question 7, why did those leaf discs begin to rise as your experiment progressed? Well, they're going through photosynthesis, right? And what happens during photosynthesis? We're producing oxygen. The leaves are producing oxygen, and that extra gas caused those leaf discs to rise to the surface. Question number 8, this one, simple point, yes or no for recording your data. Always, 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 you need to record your data neatly, accurately, so that somebody else looking at your lab manual could repeat the experiment using the information that you have. So always record your data neatly. Question number 9, did you complete that line graph? Obviously, your answer should be yes. The breakdown of points, and again, your parent, your facilitator will be looking for this. You do need to have a neat graph. That's 1 point. Your graph needs to be labeled. Your y-axis, your x-axis need to be accurately labeled. You need to have a line that accurately represents the data from the table in your lab manual. That is worth 2 points for this graph. Okay, question number 10. What process does this equation represent? That is cellular respiration. What clues you in? Well, if you look at it there, we've got C6H12O6. That's glucose on the left side, and oxygen, breaking it down, releasing that ATP, the usable energy for the cell. That lets you know it's cellular respiration producing that usable energy. Question 11, this one was worth 2 points. What about that relationship between photosynthesis and cellular respiration? Well, remember, let's start with photosynthesis. It's converting that light energy from the sun and it's storing it in the chemical energy of glucose. What chemicals are we using? We've got to have that carbon dioxide, and it's releasing oxygen. Okay, that's photosynthesis. Now let's compare that to cellular respiration. Cellular respiration is starting with glucose. Okay, photosynthesis was making sugar. We were storing it in the chemical energy of sugar. Cellular respiration is starting with the product of photosynthesis. Starting with the glucose, starting with the oxygen, and then producing the usable energy of ATP. Carbon dioxide is given off as a byproduct. So realize these two processes are interrelated. Okay, they are not exact opposites of each other. You learned about in class, there are distinctions between them, but the products of photosynthesis are the reactants for cellular respiration. They're intertwined, closely related, but not the exact opposite of each other. Question number 12. What is going to happen when you place those control group in the dark after the leaf discs have already risen back to the top? Well, remember what is happening. Those leaf discs are eventually going to sink back to the bottom because photosynthesis, at least the light dependent phase, is going to stop. And what have you done? You've put it in the dark. Okay, and obviously photosynthesis requires light. So you're stopping photosynthesis, but realize photosynthesis has stopped, cellular respiration continues, producing more carbon dioxide. The accumulation of that carbon dioxide triggers the reformation of sodium bicarbonate. Okay, I know it sounds complicated, but stick with me. Okay, more carbon dioxide because we're still doing cellular respiration. The leaves are still doing cellular respiration, building up the carbon dioxide, transforming it into sodium bicarbonate. As the sodium bicarbonate builds up in the leaves, now they're beginning to sink and fall back down to the bottom. Okay, so for question number 12, eventually they're going to sink back to the bottom. Question number 13. Question 13, was your hypothesis supported or falsified? Obviously, the answers will vary for this depending upon what factor you used, but whatever factor you used, you need to use data to support your answer, whether or not your hypothesis was supported or falsified. Similar situation here with question 14. What can you summarize regarding the rate of photosynthesis and the factor that you were testing? Again, answers will vary, but you do need to have a good, clear, using the data to explain how did this factor affect the rate of photosynthesis. And finally, question 15, what steps would you recommend to improve the experiment that you performed? Several answers that you could have put here. You may have needed to do a better job of controlling variables. You know, maybe you didn't keep the temperatures consistent enough if temperature was your independent variable. If your independent variable was pH, remember I told you those pHs need to be really, really different. Maybe they were too close together for you to see noticeable results. So those are possible answers, many other possibilities that you could have put for that question as well. Okay, this experiment was basically an indirect way of observing photosynthesis. Obviously, it would be very, very hard to observe the actual process itself. It's microscopic, it's happening inside cells, but I hope that demonstrating it, even indirectly, has helped you think through and better understand the process of photosynthesis.
Remember that in this chapter, we are talking about cellular energy because no energy equals no life. We have spent a good bit of time talking about photosynthesis, okay, how photosynthetic autotrophs take an inorganic source, sunlight, and make an organic food source, of course, that being glucose. Now we're ready to move on and start talking about cellular respiration. What is cellular respiration? And before I define it for you, I'm going to ask you what it is because you should know this is one of the cellular processes and functions you should have already memorized. So, what is cellular respiration? The enzymatic breakdown of substances, the exchange of oxygen and carbon dioxide with the environment, the breakdown of food to release usable energy for the cell, or the synthesis of organic compounds. I'll give you just a second to think this through. If you are thinking choice B, the exchange of oxygen and carbon dioxide with the environment, that is not correct. That is usually our automatic response as to what is respiration, but I am asking you what is cellular respiration, and this is the definition that I asked you to memorize. Cellular respiration is the breakdown of food to release usable energy for the cell. That's the definition that you need to get down. If you are thinking exchange of oxygen and carbon dioxide, think, isn't that respiration, Mrs. Ericson? No, specifically, I'm going to call that breathing. Now, for you as an oxygen-breathing organism, okay, for you, breathing and respiration are related. The reason that you breathe, you take in oxygen, is because you require oxygen for this process, but not all organisms do. So on a basic level, what is cellular respiration? And make sure you have this definition in your notes and that you've got it on a study card and you can memorize it, especially if you missed that lesson question, because you should have this down by now. Cellular respiration, again, is the breakdown of a food substance. And we're going to use glucose as the common example. Okay, the breakdown of a food substance into usable cellular energy. And now I'm adding to that definition a little bit because we have discussed ATP. Remember, glucose and other large organic molecules are too big. It's like trying to put a $20 bill in a vending machine. You can't get ready to use cellular energy from glucose. You need a more moment-by-moment supply, like a quarter in the vending machine, and that, as you can see on the slide, is ATP. So remember that ATP is our usable form of cellular respiration, or of cellular energy. Now, before we get too much further, I'm going to ask you another lesson question. Lots of reviewing here, okay? Now, where does it occur? We've talked about all the cellular organelles and the functions that they carry on. There's a lot of overlapping there, but when we talk about cellular respiration, where does that primarily occur? In the ribosomes, the mitochondria, the endoplasmic reticulum, or the nucleus? We think of an organelle, like with photosynthesis, we think of chloroplasts when we're dealing with eukaryotic organisms. With cellular respiration, you ought to be thinking mitochondria. Remember the mighty mitochondria, the powerhouse of the cell? The reason we call it the powerhouse of the cell is because this is where cellular respiration will primarily occur for eukaryotic cells. Remember, eukaryotic cells have membrane-bound organelles. Prokaryotic cells do not. So, for eukaryotic cells, they're going to use the mitochondria. Now let's see if we can get the big picture here. Okay, what are we doing with all these energy conversions that we've talked about? Okay, remember we have our autotrophs. They take an inorganic substance. In the case of photosynthetic autotrophs, they're using sunlight. In the case of chemosynthetic autotrophs, they're using some other inorganic chemical. We're taking an inorganic source and making an organic food source. Now, cellular respiration is kind of going to be the opposite, not the reverse, but kind of the opposite. So as we get started here with cellular respiration, let me at least give you the big picture, what you already know, so we can kind of wrap our brains around it, okay? Remember photosynthesis, and again, this isn't the full equation, but the simplified idea, the big picture. In photosynthesis, we're taking kinetic energy, sunlight. That is inorganic. And at the end of photosynthesis, we have stored chemical energy, typically in the form of glucose, C6H12O6. You ought to be saying that in your sleep by now, okay? So that's the big picture for photosynthesis. But again, that organic molecule is too big. It's too big for the photosynthetic autotroph. It's too big for the heterotroph, or for the consumer that is consuming from another organism that large organic molecule. So we have to break it down. And that's what cellular respiration is all about. Taking now that stored chemical energy, that larger organic molecule, and breaking it apart into usable chemical energy. And again, you should be familiar, that's the diagram that we use, that's ATP. Because ATP, we have 3 phosphates attached, represents stored energy. That third phosphate breaks off, you release energy, you end up with ADP. Okay, that should be good review for you. You ought to understand there what I'm talking about. So these are kind of the opposites of each other. If you notice, with photosynthesis, light energy, end up with glucose. Cellular respiration, you start off with glucose, you end up with ATP. But it's not like they're really the reverse of each other. It's not like now we're going to take those steps we learned in photosynthesis and we work them backwards and ta-da, we have cellular respiration. No, 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 no. It doesn't work that way. Just realize, though, the big picture. Okay, now we're taking that glucose, breaking it down to form usable energy, ATP. When we talk about cellular respiration, there are two basic types, and this is probably why you confuse cellular respiration with breathing, because those two types, you are aerobic, okay? You are an aerobic organism. Animals, also aerobic. Aerobic cellular respiration, by the way, sometimes they'll abbreviate ACR, so I don't have to write out the whole thing, okay? But aerobic cellular respiration, aerobe, okay? That's an aero. That's a really good word root and combining form. Aero literally means air. It refers to oxygen. So literally, these are organisms that require oxygen to produce ATP, to produce that usable energy. Opposite, okay, if you don't know that word root and combining form AN, whenever you add an A or an AN to the beginning of a word, it negates that word. So those are organisms that do not require oxygen. So anaerobic cellular respiration does not require oxygen. And again, that's a really difficult concept for us to grasp because we rely on oxygen. We know that our cells cannot go more than, minutes without requiring oxygen. But there are organisms that do not require oxygen for that process. Now I'm going to show you an equation here, okay? And again, it looks really, really big and scary, but don't panic. Because again, before we get started in the steps and all this stuff that we have to do, I'm going to try to simplify it for you. But I want you to just settle your nerves. Okay, here we go again, another process. But I want you to think through, what do I already know about this? Okay, think about what I already know, and then we can use that to build some new knowledge. So here we go, aerobic cellular respiration. Again, as I've said and said and said, we're taking glucose. And then since this is aerobic, it requires oxygen. You see our symbol there for oxygen gas, okay, O2. So we take glucose, and in the presence of oxygen, through the process that we're about to study, what are you going to end up with? Well, there's the big picture at the very end. The goal is that energy, that ATP. But in the meantime, okay, you're also going to produce some carbon dioxide. Okay, this ought to make sense to you though, right? As an aerobic organism, you know that you breathe in oxygen, and the reason you breathe is that oxygen is necessary for your cells to go through cellular respiration. But you breathe in oxygen, and when you exhale carbon dioxide, okay? That is one of the products of cellular respiration. So again, that looks scary, but that ought to be a principle that you're familiar with. And then, of course, in this process, we're going to produce some water as a byproduct. Okay, and we'll get to how that happens here in a little bit. But that's the idea. Aerobic cellular respiration, break down glucose, the presence of oxygen, really getting ATP. Carbon dioxide and water as byproducts. Now, before we get into aerobic cellular respiration, okay, we're going to talk about the steps here. What do you need to know? Well, you need to know the steps. You need to know what goes in, what chemicals go in, what chemicals come out, okay, of each of the steps. You should also know, where does it occur in the cell, or what part of the mitochondria does it occur in. You should also know how much ATP is produced. But before we get specifically to aerobic cellular respiration, the very first step, okay, the very first step, just so you know, applies to both aerobic and anaerobic, because it doesn't require oxygen. So the very first step in cellular respiration we call glycolysis. Oh, another really, really big, scary word. But again, use your word roots and combining forms. You use those word roots and combining forms, you ought to figure out what this means. I have a lesson question for you, okay? Because really you have to know lysis, right? And we've had photolysis, cytolysis, plasmolysis, lysosome. We ought to have this down, right? So just using your word roots and combining forms, what is glycolysis? Really, you just have to tell me, what does lysis mean? You ought to be coming up with choice B, splitting glucose, lysis, to loosen, to split, to break. But this ought to make sense, okay? Cellular respiration, we're taking glucose and breaking it down into ATP. The best place to start is break apart glucose, okay? That's what glycolysis is all about. So here we go, glycolysis. As I've mentioned, make sure you have it in your notes. This is the beginning for both types of cellular respiration because glycolysis does not require oxygen. So whether we're dealing with an aerobic organism or an anaerobic organism, all kinds of cellular respiration are going to start with glycolysis, but again, that makes sense, okay? You got to start by breaking apart glucose if you're going to ever end up with ATP. This occurs in the cytoplasm of the cell. So just so you know, it occurs in the cytoplasm, but when we're done with glycolysis, the products of glycolysis will get sent then to the mitochondria to continue cellular respiration. But for now, glycolysis just occurs in the cytoplasm of the cell. So let's make sure you have a good definition of glycolysis in your notes. Okay, glycolysis is the breakdown of glucose. That's the beginning product. Okay, what's the end product of glycolysis? Well, glucose will get broken down into pyruvic acid. Your textbook goes into a lot of detail into how that happens. I'm not expecting you to know that. I just want you to know we break down glucose, we get pyruvic acid, but as we're breaking apart the glucose, we also get those hydrogen ions and electrons. That's what's being pulled off as glucose is being broken apart. As I've already mentioned, the products, okay, the pyruvic acid, the hydrogen ions, the electrons, they're all sent to the mitochondria. Remember, that's the center of cellular respiration in eukaryotic cells. So the products are sent to the mitochondria. One of the other end products of glycolysis, the whole point of cellular respiration, we're going to get 2 ATP. Now notice I said 2 net ATP because with this, again, we need a little bit of activation energy to get this started. So we have to spend a little energy to make a little energy. The idea here is we've got 4, we make 4 ATP, but we spent 2 at the beginning. Don't worry about all that, but just know it's 2 net ATP. Now that doesn't seem like a lot of ATP. That's because really we're working up to making a lot of ATP. Notice I just underlined those hydrogen ions and electrons. Why are those there? What's the point? Well, with glycolysis, we're going to make a little bit of ATP. But in your notes, circle those hydrogen ions and electrons. The really important part of this, make sure you get this, as glucose is broken down to pyruvic acid, we're pulling off those hydrogen ions and electrons to be used later to make a lot of ATP. Okay, and again, those hydrogen electrons get bonded to a chemical to be saved later, all that stuff, just like we talked about in photosynthesis, but don't have to know the chemical name and all that kind of stuff. Just know we pulled off those hydrogen ions and electrons to save for later to make a lot of ATP. Now that's glycolysis. Again, breakdown of glucose to pyruvic acid, but we're getting hydrogen ions and electrons and a little bit of ATP. The next 2 steps that I want to talk to you about, these are aerobic. Okay, so there are 2 steps. Glycolysis occurs in aerobic and anaerobic organisms. But when we move to aerobic cellular respiration requiring oxygen, there are 2 steps. And again, I'm going to try to keep this really simple for you. Your textbook goes into a lot more detail, but let me give you that first step of aerobic respiration. It is called the Krebs cycle or the citric acid cycle. Now there are 2 names. Krebs cycle because that's the guy who, identified the steps. That's the guy's name. But you can see here we're going to take that pyruvic acid from glycolysis and break it down into citric acid. So that's where it also gets its name. It is a cycle. It's kind of interesting. It cycles around. You can kind of see that diagram in your textbook. Basically, we take the citric acid, okay, and it gets broken apart, but it gets recycled back into citric acid. So what's the point of that? Well, the point is hydrogens and electrons, because we're going to use those later to produce more ATP. Okay, in our next lesson, this is where we're going to pick up. But study, 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 make those study cards, and Lord willing, I will see you next time.